Thanks a lot for your kind invitation to present our recent work published in Science. So basically, we discovered that a gaseous signaled ethylene is essential to sense the level of soil compaction. This multidisciplinary work could not have been possible without this Hounsfield facility. This provided us to look into the soil structure and how change in soil structure inhibits the gaseous movement. This Hounsfield CT imaging facility brings together the soil scientists, root biologists, mathematical modelers, cell biologists, engineers, and the computer scientists to underpin some of the most complex biological mechanisms underlying plant root growth behavior in different agroclimatic conditions. This is the LEGO model of our CT imaging facility. So we have three different types of CT scanner to image from nanoscale to whole plant scale. So basically these three CT scanners are nanotome, mid CT scanner and large CT scanners. Nanotome can provide very high resolution up to 1 micron, and the large CT scanner, which is integrated with the robot, can generate the images of entire life cycle of different crop species. So in this video you can see the moving robot holding a large crop plant in the growth facility. This robot can directly enter into a room-sized CT scanner, and this large CT scanner allows us to image nearly 1 meter long root system architecture which is grown in real soil conditions. So this is the actual X-ray screen where plants continuously face the X-rays from bombarded from the X-ray guns, and it takes nearly 8,000 images of the 1 meter long root soil column, and this produces huge data set in terabytes, and these images are then reconstructed to visualize the 3D images of root system architecture in soils. So we employed this non-invasive root imaging system to monitor the root adaptive responses in compacted soil conditions. Without this imaging facility, it would have not been possible to dissect the mechanism underpinning root adaptive responses in compacted soil. And this facility played a very instrumental role to address the problem of soil compaction. Soil compaction is a modern agricultural problem which relies on the use of heavy machinery such as big tractors and harvesters. These big farming equipments basically compress the topsoil as well as the subsoils by the heavy beds. Thus, compacted soil does not allow the root tip to penetrate harder in the soil, and basically it prevents the foraging capacity of the plant, and thus plant faces severe yield reductions in compacted soil, and this yield reduction can be as high as 75% when combined with the drought. Therefore, soil compaction is a massive challenge, and it is spreading very fast that nearly two-thirds of the entire European agricultural soil is prone to soil compaction. To understand this plant root problem, we started to investigate that how compacted soil inhibit the root growth. So this is a CT image of rice roots growing in non-compacted and compacted soil. And you can clearly see that the soil is fluffy and not imposing any mechanical impedance in 1.1 bulk density soil, which we say the non-compacted soil, and the roots can nicely grow throughout the column. However, when we compacted the soil up to 1.6 bulk density, the rice root tips struggled and the root growth was inhibited up to 50%. We have also examined the root length in the uprooted plants from the non-compacted as well as the compacted soil, and you can clearly see that as we increase the compaction level, root tip growth is progressively reduced. And this root growth inhibition blocks the ability of the roots to acquire the nutrient and water and which support the plant growth, and eventually this negatively impact the crop yield. What are the root adaptive responses of a plant growing in the compacted soil? Well, one of the classical root adaptive response is exhibiting radial expansion of the root cortical cells, and which results into the thicker root in compacted soil as compared to a non-compacted soil. And this striking response is mostly present in root elongation and root differentiation zone. This root adaptive response provoked us to identify the signal or the growth regulator which is causing this radial expansion response in compacted soil. To discover this signal, we found that ethylene treatment causes the same root growth inhibition and radial expansion of the cortical cells as if the roots were growing in the compacted soil. To confirm the signal, we employed genetic approach by examining the root growth responses of ethylene insensitive rice mutant IN2. To our surprise, we found that ethylene insensitive IN2 mutants are blind to sense the soil compaction, and they keep pushing and penetrating the hard soil. This was quite a remarkable finding for several reasons. First, this countered the dogma in the mechanosensing field that roots are not able to penetrate the hard soil as mechanical impedance is too high to allow the root to penetrate. But this new result clearly demonstrates that ethylene insensitive mutants are clearly able to penetrate the compacted soil. Moreover, this also suggests that roots decide not to grow in the compacted soil, and ethylene appears to play a critical role in this decision-making process. So we were interested to know whether this is also the case in other plant species. So to address this question, we selected Arabidopsis ethylene insensitive mutant ETR1. So like rice roots, wild-type Columbia 0 roots also exhibit radial expansion of the cortical cells in compacted soils, and they fail to grow in highly compacted soils. However, the ethylene insensitive mutant ETR1 in Arabidopsis grew normally without any radial expansion of the cortical cells. So this provides a very solid evidence that ethylene acts as a conserved signal to sense the soil compaction, particularly in rice and Arabidopsis. So, what is the relation of ethylene and soil compaction? Does exposure to soil compaction trigger an ethylene response in root tissues? And whether the ethylene response is enhanced in compacted soil? To answer this question, we took advantage of Arabidopsis ethylene reporters called IN3GFP. So this ethylene reporter is stabilized after the exposure to ethylene, and you can visualize the enhanced GFP signals in the root nuclei. So when we treated this reporter with a light dose of ethylene for 30 minutes, we got massive expression of these IN3 GFP signals in root nuclei. Therefore, we took advantage of this reporter to see what happens in the compacted soil. We also quantified this fluorescence signal after the ethylene treatment, and which was significantly very high, uh, as you can see in the bar graph. So we took advantage of this iron 3 GFP reporter and grew them in non-compacted soil. To our surprise, we didn't see any stabilized GFP signal in non-compacted soil. So effectively, the reporter didn't produce any ethylene signal of, under non-compacted soil. However, when we grew them in compacted soil, they exhibited clearly enhanced and stabilized GFP signal in root nuclei growing in compacted soil. And if you remember, exactly this is the same elongation zone where we see most of the radial expansion of the cortical cell and epidermal cell reduction or expansion under compacted soil condition. So this was really a very exciting result, clearly demonstrating that ethylene can act as a key signal to sense the soil compaction. To confirm this Arabidopsis reporter results, we asked our Chinese collaborator Dr. Gujiang Huang to create similar ethylene reporter lines in rice. Professor Dabing Chang and Dr. Gujiang Huang are our superb collaborators from Shanghai Jiaotang University, China, and this type of a very strong collaboration ensures to achieve our common goals and share the success. So we employed this rice reporter line to grow them in compacted soil, and interestingly we found similar enhanced expression of IL-1 GFP in highly compacted soils. So this begs a question: how does ethylene response increase during a root compaction response? A enhanced ethylene response can be explained by two mechanisms. The first one is either somehow mechanoperception is causing elevated level of ethylene biosynthesis or signaling. To confirm this, we dissected the rice root tips and profiled the ethylene precursor level ACC in both non-compacted as well as compacted rice root tips. To our surprise, we didn't find any statistically significant difference in ACC level in root tips of compacted or uncompacted soil grown rice root tips. However, in compacted soil condition, the soil pores get collapsed and become very constricted, which restrict the gaseous movement, and this blockade the ethylene diffusion near the root tips, and which results into local accumulation of ethylene, which eventually trigger the enhanced ethylene response. So we tried to resolve this hypothesis by using our CT imaging facility. And so Dr. Craig Streck, who is an expert in the CT imaging facility, um, he performed the soil pore analysis of the non-compacted and compacted soil, and this analysis revealed that indeed the soil pores become almost nil in compacted soil as compared to non-compacted soil. Thus, diffusion of gaseous hormone ethylene might be very slow in these very small air-filled pore spaces. To validate this assumption, we performed mathematical modeling of ethylene diffusion in these soil pore networks in non-compacted as well as in the compacted soil conditions. Our collaborator, Professor Olivier Martin, simulated the model and predicted that indeed the diffusion of ethylene is severely restricted in compacted soil condition than non-compacted soil condition. And you can clearly see the difference of ethylene diffusion at green line versus red line. What they did was a very simple and elegant experiment. Two airtight chambers, the top chamber and the bottom chambers, were connected with a neck where we can place our non-compacted or compacted soil columns. So Son injected the ethylene in the upper chamber, and subsequently he collected and measured the ethylene level in both the upper and lower chamber. So what we found was a very remarkable. In non-compacted soil, it took nearly 100 minutes to reach from upper chamber to lower chamber. However, in compacted soil, it never reached in the lower chamber till the completion of the experiment, which took nearly 20 days. So finally, we conclude that ethylene diffusion is clearly blocked in compacted soil condition. So the model which we predicted earlier was entirely and absolutely right. Based on our CT image of soil structure, pore networks, and mathematical modeling, and ultimately the experimental validation, we confirm and conclude that in compacted soil condition, ethylene is clearly blocked, and its accumulation near the root tip enhances the ethylene response in compacted soil, and which basically shape the root adaptive responses in compacted soil condition. So isn't this model appears so simple that by mere blocking the diffusion of ethylene can really trigger the ethylene response? So to prove this, we designed a very simple and ingenious experiment to answer this question. So what we did, we covered the root tips with a gas impermeable barrier for 2 hours, which will effectively prevent most of the diffusion of the ethylene from the root tips. So we performed this experiment using our Arabidopsis IN3GFP reporter and simply covered the root tips with the high vacuum silicon grease. To our surprise, we found that the GFP level is incredibly increased even after the 2 hours of the application of the gas impermeable barrier. So this elegant experiment confirmed that blocking the ethylene diffusion is clearly able to enhance the ethylene response. So we also know that hypoxia can also induce the ethylene response. And to dissect the role of hypoxia in compacted soil condition, we took advantage of several hypoxia reporter such as PCO and GFP gas reporter line and grew them in compacted and non-compacted soil conditions. So unlike in the submerged condition, which act as a positive control for hypoxia reporter, we didn't see any enhanced expression of hypoxia reporter in either the compacted soil condition or by blocking the diffusion of ethylene using gas impermeable barrier on half ms agar plate conditions. So this experiment clearly ruled out the possibility of hypoxia-associated enhanced ethylene response in compacted soil condition. So we performed all the previous experiment in sandy loam soils, and it's a very general and common idea that whether this mechanism operates across the different soil types. So it's important to understand that there are several thousand types of soil, and it's very challenging to do the experiment in all soil types. And therefore, we very carefully selected clay soil type, which is one of the three major soil type as recommended by our collaborator Professor Sasamuni. And I must say this, that Professor Sasamuni went to the bunny field and collected the clay soil during COVID lockdown And we performed this experiment in clay soil by using our new rice-based ethylene reporter. And this experiment clearly revealed that this restricted ethylene diffusion-led enhanced ethylene response is conserved in different soil types. So to summarize this seminal discovery, this video illustrates that under non-compacted soil condition, ethylene is easily diffused away using the large air-filled pore networks and doesn't pose any restriction for root growth. As you can see that both wild type and ethylene insensitive mutants are happily growing in the non-compacted soil. However, in compacted soil condition, only the ethylene insensitive mutants is able to penetrate the hard soil, whereas the wild type root really struggles to do so. So, in summary, soil compaction is a major agriculture problem, particularly the modern agriculture problem, which negatively impact the crop yield and resource uptake. And our results clearly reveal that root growth arrest is an adaptive response rather than just caused by the physical limitation. And this new signaling paradigm where gaseous diffusion of ethylene plays a critical role, which enables the plant roots to sense the soil compaction directly. And it also showed that disrupting plant ethylene sensitivity can enable the plants to penetrate highly compacted soil. And our discovery opens new avenues to breed crops having roots with reduced sensitivity to compaction, and we can create the future-proofing crops which can withstand the highly compacted soil conditions. So our noble insights were only possible by working with other disciplines such as soil science, advanced imaging, and mathematical modeling. And finally, I would like to say a big thank you to all of the key collaborators, reviewers, and many experts who played very critical roles to improve the manuscript and our understanding of ethylene diffusion and soil compaction responses, which prevent the root tip growth and thus blocks the ability of the plant root to forage for nutrients and moisture, and ultimately crippling the yields of the crop. Particularly Professor Dabing Chang and Dr. Guxiang Huang, who works very closely with our team and supported the work by providing most of the key rice genetic resources in this work. And they played very instrumental roles in this work. Doctor Craig Sturrock, who is an expert in CT imaging, played very critical role and essential roles to image the soil pore networks in compacted and non-compacted soil. Dr. Rahul Bhosle, who initially involved in this project, really catalyzed us to dig deeper in the soil compaction project. I would also like to say thank you to Loti, and she was a master student and really helped me to image the cortical expansion of the rice roots under compacted soil conditions. And I must credit to Dr. Sean Hartman and Professor Renz, the very talented and expert ecophysiologist who played a very critical role to measure the diffusion of the ethylene in compacted and non-compacted soil conditions. And I really enjoyed working with Professor Olivier Martin and his inquisitiveness, which always fascinated me. So he performed the mathematical modeling to predict the rate of ethylene diffusion in compacted versus non-compacted soil based on the real soil pore image data of the CT images. I would also like to acknowledge Professor Karen and Dr. Michael Karati, who clearly helped us to measure the ACC level in the rice root tips grown in non-compacted and compacted soil conditions. And I must say a big thank you for your guidance and support Professor Richard Bailey, who is a soil scientist and who helped immensely to perform the measurement of the physical properties of the non-compacted and compacted soil conditions. And I would also like to say a big thank you to Professor Jonathan Lynch and Professor Kathy for their support and intellectual contribution to shape our story to reach at this stage. And I must say that it could not have been possible without the constant input and critical support by Professor Sasa Muni, who enlightened us in every meeting regarding the soil compaction responses. And finally, Professor Malcolm Bennett, who was the main architect to bring together all the key peoples who immensely supported and contributed to conclude this successful research project. His excitement toward the novel research and interdisciplinary finding always encouraged me to propel this kind of highly multidisciplinary research. His role in shaping this story is really unparalleled. And finally, we are very grateful for all of the funding from different agencies such as ERC, Royal Society, BBSRC, EPSRC, Beacons, and excellent support from Nottingham University, who played a very important role in this research work. And finally, thanks a lot to all of you who are actively listening this webinar. Finally, I would like to reap this opportunity to invite all of you in Routing 2021 at 9th International Symposium organized by University of Nottingham in collaboration with University of Missouri, which is parallelly organizing 11th Symposium of the International Society of Root Research. The registration is open now. Please go to the Routing 2021 website and register your participations. To encourage maximum participation, the registration is free for developing countries, and we welcome all of your participation in Routing 2021 Symposium. We have also organized special themed issues in journals such as Frontiers in Plant Science, Physiology of Plantarum, and Plant Cell Environment. Please note the registration date, which is open now, and the cost of registration is very minimal and competitive. We are very excited to see you in the meeting. And finally, thanks a lot to all of you for joining me in this talk. Thank you very much.